Hi, everyone. My name is Anjani, and I'm a conference director for Cambridge Health Tech Institute. I'm also the lead director for our drug discovery chemistry event, and that happens every April in San Diego. This year, it's our 20th annual, and one of the tracks that we've had there from the beginning, so it's a multi-conference track event, and one of them that's been there from the beginning 20 years ago and is still there today is our fragment-based drug discovery conference track. So obviously that's celebrating its 20th edition as well. So this year, to mark that occasion and kind of, learn more about what has made fragment-based drug discovery so successful over all these years. I've invited Stephen Fesik, who's a professor at Vanderbilt University and considered the founder, one of the founders of the field, to be our speaker. He has spoken for us before as a plenary keynote, but this talk will be different. We want to hear about what has enabled his success ability to bring quite a few molecules, through clinical trials into the market. So I have Steve here with us today. Thank you for spending the time, and he's going to tell us a little bit about what to expect at his talk and how it's going to be different from some of the previous talks he's given for us. So, Steve, I'll turn it over to you now.
Well, thank you. And it's true. I've often get asked to talk about particular applications of the method, but rarely do I get the chance to talk about the method itself. And we have been using this for almost 30 years. It's hard to believe, but yes, that's true. And I'd like to tell you a little, and this talk is going to be largely geared around the method, and we'll break it down to different techniques, what's important, what's not important. And for example, I'm going to talk about the methods that we use, the approaches, and the best practices. In detail, I'll be covering things like how do you build the optimum fragment library for doing the screen? I'll be talking about how do you optimize what's important in optimizing protein preparation to do the screen. I'll cover how we do the NMR method, and if we use looking at larger proteins, we use C13 methyl labeling, but we use TROSY-based techniques, N15 proton correlation maps. Depend on the problem that you have, but it's always protein observed, and then I'll talk about the advantages of that. Then, after you get hits in the screen, I'll talk about another very important thing. Like a lot of people can get hits. What do you do with them? They're usually weak hits, and I will go into how you can optimize them using fragment linking, fragment growing, or fragment merging. And I'll provide examples of this, how we've used each of these techniques of how to elaborate the molecule, how you change the hits that you get to leads.
Great. So it really will be focused for people practicing fragment-based drug discovery, it sounds
My
like.
goal is when you get through with my talk, you'll be able to go out and get drugs like I have from using, if you follow the recipes that I give.
Get drugs to market, you mean?
Yes.
And what are some of those that you've
Well, we've targeted our particular approach, and one of the advantages of fragment-based drug discovery is that it allows you to go after really difficult targets, targets that industry largely shied away from or thought they're too difficult to go after. But yet, some of these targets are highly validated. So, we have gone after RAS inhibitors with Boehringer Ingelheim. We have gone after MYC In a lot of ways, we've tried to target MYC, and we've tried to target the Wnt pathway to drug that. These are highly validated targets, but they have one thing in common. The protein that you'd like to inhibit is thought to be undruggable. And so this is right up my alley. This is what we focus on. And that's what and using these techniques, you can get traction in this area.